Hi there, and welcome to this video on A-Level Biology for the AQA specification, focusing on the topic of cells, and in particular, cell organelles. I'm Manisha from StudyMind, where we help you to revise A-Level Biology with our helpful video tutorials tailored to your subject, your specification, and to you. If you're new here, please make sure to click that subscribe button. And whilst you're watching, feel free to leave any comments down below of anything you're unsure about, and let us know if it's your first time watching so we can send you our free revision resources. We also have helpful timestamps to guide you through the specification. So, let's get started. Welcome to lesson 2 of 9 in this tutorial, covering the topic of cell organelles. This is our second video in our series of 9 lessons on the topic of cell structure. In the last lesson, we looked at the similarities and differences between eukaryotes and prokaryotes. Here are the key learning objectives for today's lesson. First, we will look at animal and plant cells. And then we will look at cell specialization and organisation. Here are the AQA specification points for this tutorial. Feel free to pause the video now and have a read through them before we begin. The first one is to deal with the structure and function of various organelles. Here are the 11 substructures in cells we will be covering in this section. We can see all the organelles of a cell shown here. Let's start with the cell membrane. The cell surface membrane is also commonly called the cell membrane or the plasma membrane. It always provides the cell with structure and is made up of a phospholipid bilayer with proteins regularly embedded along its length. It can also have lipids and carbohydrates embedded in or on it. It helps to regulate the movement of substances into and out of the cells. It can also be a barrier to protect the cell from the external environment. Here's a summary of the cell surface membrane structure. The cell surface membrane can help with extracellular communication or communication between cells. It has special proteins called receptors, which are glycoproteins and glycolipids, which can receive signals, such as hormones, from neighbouring cells or from the environment. They relay these chemical messages to the inside of the cell to initiate specific responses related to these messages. Now let's look more closely at the nucleus. All eukaryotic cells will have a nucleus. The nucleus houses the genetic information of the cell. DNA is packaged in the form of chromatin, which in turn is packaged into chromosomes. These chromosomes are stored in the centre of the nucleus. The nucleus has its own membrane. This membrane is a double membrane, which is also called the nuclear envelope. Like the cell surface membrane, this membrane also regulates what gets into and out of the cells. The nucleolus is the region of the nucleus where ribosomal RNA is assembled into ribosomes. There may be multiple nucleoli. The nucleus controls all the activities of the cell. Because the nucleus houses the DNA, the nucleus also regulates all cellular processes. It does so by not only regulating the replication of DNA, but also by regulating the process known as DNA transcription. This is the process by which the genetic information stored in the DNA is turned into RNA, which in turn can be made into protein by ribosomes. You're unlikely to get a 10-mark question asking you just to list the functions of all organelles. Instead, you're more likely to get a 3-mark question, focusing on the functions of one specific organelle, such as the nucleus. Now let's look at the mitochondrion. The mitochondrion is a membrane-bound organelle. It's made up of 2 membranes, an inner membrane and an outer membrane. The inner membrane is folded to create cristae. The inner region of the mitochondrion, located inside the inner membrane, is called the mitochondrial matrix. The mitochondrion is the powerhouse of the cell. It is where aerobic cellular respiration and ATP synthesis take place. The mitochondrial matrix contains enzymes for cell respiration. Mitochondria have their own DNA and is similar to bacterial plasmid DNA. Not much is known about it and its function in the cell, or even in the mitochondria itself. They also have their own ribosomes, yet, like mitochondrial DNA, not much is really known about its function. We assume the ribosomes are for making mitochondria-specific proteins from genetic information stored in the mitochondrial DNA. The cristae enable the mitochondria to have compartmentalization. The cristae separate the mitochondria into compartments, which can each have different enzymes for different steps of the respiratory cycle. Now let's look at the Golgi apparatus. The Golgi apparatus is a stack of curved membranes. When observed under a microscope, many people describe them as a stack of pita bread. The Golgi apparatus packages lipids and proteins to be exported outside of the cell or to its surface membrane. This is why many people refer to it as the post office of the cell. The Golgi apparatus has 2 faces. The cis face receives proteins and lipids from other organelles. The trans face releases the proteins or lipids after packaging. The cisternae and the inner lumen of the Golgi apparatus will do the packaging. Proteins and lipids are packed into Golgi vesicles or membrane-bound parcels. They are essentially envelopes for transporting the lipids and proteins to their final destinations. The Golgi apparatus also makes lysosomes. There are 2 easy ways for you to spot Golgi apparatus in an exam. First, it will have vesicles, forming and moving around its edges. Second, it usually appears curved and smooth. Next, we will look at lysosomes. Lysosomes are round, membrane-bound organelles. They have no distinct internal structure and they're a type of Golgi vesicle. Lysosomes contain enzymes which are called lysozymes, which digest material in the cell. The membrane surrounding the lysosome protects the rest of the cell from their digestive activity. Lysozymes are known as hydrolytic enzymes. They are essentially the waste disposal system of the cell. They are used to digest food, organelles, and other cells. Lysosomes can be used to digest invading cells or worn-out cell structures. They can ingest the broken down organelles through endocytosis and digest them using lysozymes. Lysosomes can do the same thing to invading pathogens, such as bacteria. The digested material can be used by the cell as building blocks to make other molecules and structures. Alternatively, it can be removed from the cell as waste. Now let's look at ribosomes. Ribosomes are tiny and they're often shown as dots in the cytoplasm. They're made up of 2 subunits, a small one and a large one. They are found floating freely in the cytoplasm. They're not membrane-bound organelles, but some of them can be bound to the endoplasmic reticulum. They are made up of rRNA and proteins. Hence, they are a type of ribonucleoprotein. They read the mRNA and use the instructions stored in it to make proteins. Ribosomes receive genetic information from the DNA in the form of mRNA. The rough endoplasmic reticulum contains stacked, thin, and fluid-filled membranes. It's structurally very similar to the Golgi apparatus. It's a membrane-bound organelle located near the nucleus. Next up, we'll cover the rough endoplasmic reticulum in a bit more detail. Here, we can see the rough ER in contrast to the smooth ER. The RER surface is covered in ribosomes. This is why we call it the rough endoplasmic reticulum, because it looks bumpy under a microscope. The ribosomes attached to the RER are responsible for making proteins to be exported outside the cell or to its membrane. We can see the rough ER here. Proteins meant for export are made by the ribosomes and then released into the RER, which folds them and packages them to send them to the Golgi apparatus. Now let's look at the smooth ER. The SER is structurally similar to the RER, except it's smooth because it doesn't have any ribosomes on its surface. The SER synthesizes many lipids, such as cholesterol, and other molecules such as steroids and hormones, for use inside the cell and for export outside of it. When exporting these molecules, the SER packages them and sends them to the Golgi apparatus. The SER plays a crucial role in the detoxification of toxins that are either consumed by the cell, such as alcohol, or toxins produced as byproducts of metabolism. Now, we'll look at some plant-specific organelles. The first of these is chloroplasts. This is a chloroplast. As you can see, there are various parts to it. Chloroplasts are found in algae and plants. They are flattened, membrane-bound organelles. They have inner membranes, which we call thylakoid membranes. They are small, thin, and are often stacked into structures that we call grana. The singular of these is granum. These grana are connected to each other by thin pieces of thylakoid membrane called lamellae. Stroma is a thick, viscous fluid that fills up most of the chloroplast. Photosynthesis involves several reactions. Some are light-dependent, others are not. Different parts of photosynthesis take part in different parts of the chloroplast. For example, light-dependent reactions will take place in the chlorophyll. Some parts of photosynthesis will take place in the chlorophyll, whilst other parts will take place in the stroma. If you're asked to label a diagram of a cell, you can sometimes spot the chloroplasts easily because they have stacks inside them, representing grana. Next, we will look at the cell wall. The cell wall is found in plant, algal, and certain fungal cells. The cell wall is very strong and is made up of a carbohydrate polymer called cellulose. In fungal cells, it is made of chitin. Some bacteria can also have cell walls. Its main function is to support the cells, but it also offers a small level of added protection. It gives the cell structure,
preventing it from changing shape. Unlike the cell surface membrane, which forms a phospholipid barrier, it is freely permeable. It's permeable to all soluble substances which can pass through the small pores in the cell wall. Now let's look at a vacuole. Plant cells have vacuoles. They are surrounded by a membrane which we call a tonoplast. A few animal cells can also have vacuoles. It mainly contains a cell sap. This is a store of chemicals and mineral ions, including salt and sugars. A vacuole helps to maintain the pressure inside a plant cell by pushing the cytoplasm against the cell wall. This can prevent the plant from wilting. The vacuole will isolate harmful chemicals from the rest of the plant cell. The tonoplast separates the contents of the vacuole from the cell cytoplasm. Let's consider some of the differences between animal and plant cells. Plant cells have chloroplasts for photosynthesis. Animals do not undergo photosynthesis. Secondly, plant cells have a cell wall. Animal cells only have a cell surface membrane. Finally, plant cells have a large central vacuole, whilst most animal cells do not. Next, we will look at the specializations of cells. In multicellular organisms, cells specialize to perform specific functions. Specialized cells will look different from each other, as they can adapt by changing shape and composition of the organelles. For example, red blood cells adapt to transporting blood. A specialized cell can have one or more organelles relative to other cells. Therefore, cells with different functions oftentimes look drastically different to each other. Here are some specialized cells you should be aware of. We will cover epithelial cells again in another video, but we'll mention it briefly here. The intestine has many villi, used to absorb nutrients that have been digested. These will have a thin wall, allowing for a short diffusion distance, a rich blood supply to maintain a concentration gradient, and a large surface area to aid the speed of diffusion and active transport. There are some microprojections as well, called microvilli. These will increase the surface area further in order to absorb more nutrients from the digested food. The epithelial cells will have a large number of mitochondria. Absorbing and transporting nutrients requires a significant amount of energy. Now, let's look at red blood cells, which are also known as erythrocytes. These will transport blood and oxygen across the body. They have no nucleus to increase their storage capacity for hemoglobin, which binds to oxygen inside the cell. Red blood cells do not have DNA, and they do not divide or carry out most other typical cell functions. Sperm cells carry genetic information of the male to the egg of a female during sexual reproduction. They have many mitochondria to provide energy to travel to the long distance to the egg. They have a tail which helps them to swim through the vaginal canal and into the uterus. Let's look more closely at the structure of a sperm cell. First, they have an acrosome, which contains the enzymes necessary to digest the outer layers of the egg cell. There is also a long tail, which helps them to swim through the female reproductive system. They also have mitochondria, which provide energy for movement. Importantly, they have a big nucleus to hold the genetic information. Hepatocytes are liver cells. One of the main functions of the liver is to carry out detoxification, which is helped by the large, smooth ER. Our final specification point is to look at the organisation of these cells. Cells develop as the organism does. They are adapted to perform a specific function. These cells alone, or with other specialised cells, can form a tissue. For example, epithelial cells make up epithelial tissue. These can make up glandular tissue as well. Muscle cells make up muscular tissue. Neurons make up nervous tissue. And blood cells make up hematopoietic tissue. Different tissues and cells can group together to make an organ. Here are some examples. Different organs with similar functions group together to form an organ system. The digestive system is made up of the mouth, esophagus, stomach, small intestine, pancreas, gallbladder, and the large intestine. Different organ systems group together to create an organism. Humans have many different organ systems. Some examples of the systems in the human body are shown here. These are the most common A-level biology examples of specialised cells, so they are definitely worth learning. Now try and label this diagram. Here are the answers. Let's fill in this table as a summary of all the information we've just learnt. First, we'll go back to the cell surface membrane, which was a phospholipid bilayer. Its function was to regulate movement into and out of the cell. Next, we will look at the nucleus. This consisted of a nuclear envelope and the nucleolus. Its function was to carry the genetic information and to control the cell activity. Now we will look at the mitochondrion, which consisted of cristae, its own DNA, and ribosomes. Its function was to help with respiration. Now let's look at chloroplasts. This contained thylakoid membranes stacked into grana. It also had lamellae and chlorophyll. It was used for the light and non-light dependent reactions of photosynthesis. Next, we'll look at the Golgi apparatus. This had stacks of membranes which had cis and trans faces. Its function was to receive lipids and proteins and package for export. It also made lysosomes. Now let's look more closely at lysosomes. Lysosomes contained lysozymes. And these were used to digest pathogens and worn-out cell structures. Now, let's look at ribosomes. These were made of two subunits floating in the cytoplasm. There was one large and one small subunit, and these were used to make protein using mRNA. Now let's look at the rough ER. The rough ER was covered in ribosomes. It was used to package proteins. Next up, we have the smooth ER. This had no ribosomes. And it was used for lipid synthesis and detoxification. Now let's take a look at the cell wall. In plant cells, this was made of cellulose. Whilst in fungal cells, it was made of chitin. Its function was to be strong and support cells. Finally, we'll look at a vacuole. A vacuole was made up of cell sap and a tonoplast. It was used to maintain pressure and to store salts and sugars. We've now covered all the specification points for this lesson. Feel free to pause the video now and re-watch any of the sections you feel unsure about. We've now completed Lesson 2.
If you enjoyed this tutorial, make sure to subscribe by clicking down below and leaving a comment of the topic that you'd like to see a video on. Click here to watch the rest of our videos in our A-Level Biology series, or visit our website, studymind.co.uk, for past paper compilations by topic and specification.